Welcome to Azad Bioacademy. So now come to the next stage, that is second stage, that is membrane stage. Another name is called elongation and transmigration stage, or simply you can say elongation and transfer stage. So for easy understanding, I will draw here the membrane. That is, that is the lipid bilayer, and this is the bacterial plasma membrane. And this is called membrane stage because membrane will carry different types of enzyme, and that will contribute to convert the sugar that already we got inside the cytoplasm, that is NaCl, UDP, NaCl, uramic acid, just which was carrying pentapeptide. Okay, so that will convert into the another form, and it will be associated with another sugar. Okay, so now what will be happening here? Now I will just try to explain. So in the membrane, it will carry specific types of lipid, and that lipid, carrying initially one phosphate. Okay, so this lipid name is called bactophenol. Bactophenol. You can say also C55P. That is, the carbon 55 number of carbon containing lipid, and which is carrying one phosphate. So C55P. You can say simply lipid P. Okay, lipid phosphate. Or in another chemical name is caprynyl phosphate. Okay, so different name. But for your easy understanding, several times I will try to use the terminology bactoprenol. Okay, so now this is the bactoprenol, and that bactoprenol initially it is carrying on phosphate. Okay, so this is the bactoprenol monophosphate containing bactoprenol. That bactoprenol, and here another enzyme is there. You can think this is another enzyme. This is called MRAY. Okay, and this on the function of the enzyme is to attach the N-acetyl muramic acid containing pentapeptide containing N-acetyl muramic acid to that bactoprenol, because bactoprenol is very important lipid to just carry that the building block of the peptidoglycan, and finally it will help to flip up the basic unit from inside to outside. So come here. So that will add that the monomer previously formed unit was UDP. That was UDP, N-acetyl muramic acid carrying pentapeptide. Already I mentioned by different color, that is, L-alanine, this is D-glutamine, this is L-lysine, and this is the dimer of D-alanine and D-alanine. Okay. So, in the earlier stage, if you just recall, then you will get the exact figure. And that will attach with that bactoprenol by the help of enzyme MRAY. Okay, how it will attach? That is here, UDP here, UDP. So that UDP, that means uridine diphosphate. So what will happen? Uridine 1-phosphate will flip out from here, and 1-phosphate will attach here. So you can add the phosphate from here, and it will be attached to the sugar. Okay, so finally what will happen? That stage I will show you here. So this is the bacterial, and this is the diphosphate, and that is the sugar unit. That is the N-acetylmuramic acid, and that is the pentapeptide. Okay, pentapeptide. So now this pentapeptide containing sugar is attached with the bacterial by diphosphate. Okay, so this is called lipid 1. Okay, lipid 1 means that pentapeptide containing N-acetylmuramic acid is completely attached with the bacterial by 2-phosphate. Initially, it was carrying the bacterial carrying 1-phosphate, and now it is in that form. Okay, and now another enzyme available there. That enzyme name is MORG. Okay, the function of that enzyme will just add another sugar. Already, if you just recall, that was N-acetyl glucosamine. Okay, so then that N-acetyl glucosamine will add here by the help of that enzyme. So now you can add bacterioprenol. This is diphosphate. This is your muramic acid, and here another sugar will add, that is the N-acetyl glucosamine by the glycosidic bond, and that muramic acid will carry the previously attached pentapeptide. Okay, pentapeptide. Actually, this is the lipid 2. Actually, this is the building block of peptide glycan. This is actually the building block of peptide glycan. But some species of bacteria also carry pentaglycine. That means additional 5 number of glycine just attached with the third number of amino acid, that is L-lysine. Okay, so here you can consider the special type of bacteria, that is, you can say Staphylococcus aureus, that will carry pentaglycine. But if you think E. coli, it does not have any pentaglycine. Okay, so I will explain in the later one what is the function of pentaglycine, and without pentaglycine how it will form peptide glycan. So now some enzyme will work here, that is FEMX, FEMA, FEMB. That enzyme, the function is to add glycine, that is single glycine will be added by that enzyme, a double glycine and then double glycine will be added where it will add pentaglycine here. So if you just add single, that is one glycine by X, then double glycine by A, FPMA, then by B, another two glycine. That is the pentaglycine. Okay? So, in a sense, some bacteria, species of bacteria, you can say this is the building block of peptidoglycan. Okay? Now, what will happen? Now, another enzyme which is present in the membrane, that is a single enzyme. Another enzyme is actually two enzymes, but those enzymes work together and function like a single enzyme. The name flippage enzyme, that basic unit will transfer from inside to outside. That flippage enzyme main function. So what will happen here? The basic unit containing the bacterioprenol, it will completely exit from inside to outside. That means it will cross membrane through by the help of flippage enzyme. It is flippage. Okay. So now you can draw here outside. So that would be the, like this is your bacterioprenol. This is containing one phosphate and another phosphate, and now it will be attached here like this is the N-acetyl muramic acid, and then this is the N-acetyl glucosamine, and it will carry pentapeptide. One is Ella, second one is D-glutamine, third one is L-lysine, and then D-alanine and D-alanine. Okay, so this is the building block or basic unit of the peptidoglycan. Now what will happen? This is phosphate and the bacterioprenol. The number of bacterioprenol is very much limited within the bacteria, so it is very much important for the bacteria of the membrane. Actually, the membrane enzyme, the main function is very crucial to just flip out the basic unit. So what will happen? The number of bacterioprenol is very limited, so it will be recycled. How it will be recycled? It will be recycled by an enzyme that is called phosphatase. Phosphatase. You know the function of phosphatase, that means it will dephosphorylate. Okay, so from here, phosphate will just out, and one phosphate carrying the bacterioprenol will again embedded to the initial stage, embedded to the membrane. Okay, so now it will free, that means it will be like that. So here you can now draw here, like that is the bacterioprenol, and it is carrying single phosphate, and then it will attach here again. Okay, so you can omit this phosphate here. So this is the unit of building block unit. Actually, the pure, that is the complete unit of the peptide glycan, that is cell wall, like a brick, no cement, no sand, that is a brick. Okay. So now we will discuss the third step, that is the extracellular step. So I will just sum up very quickly. That means within the cytoplasm, the N-acetyl myramic acid containing pentapeptide will attach the bacterial kernel by the help of MRY enzyme, and then it will form a lipid 1. And lipid 1 will convert it into lipid 2 by the attachment of another sugar, that is N-acetyl glucosamine, and that will form the lipid 2. And that lipid 2 is the basically building block, but some species of bacteria will also carry the pentaglycine, and that pentaglycine containing unit will flip out by the help of enzyme flippage, and that is the enzyme, the basic enzyme name is MURJ, and this one is FTSW, the two enzyme combinedly the flippage activity. Okay, and here the lysine containing pentaglycine. Oh, sorry. Okay, so this is all about the membrane stage, or elongation and transfer stage. Okay, so remember the flippage enzyme basically MURJ and FTSW.